Welcome to Falcon online course. My name is Manu Chakravarti. I have a PhD in physiology and cell biology and board certified in endocrinology and internal medicine. Today we'll cover endocrine physiology, and the lecture is broken down into several modules. In the first module, we'll cover some basic concepts of endocrine physiology, particularly feedback loops and some basic concepts about how hormones work. And then in the subsequent sections, we will be covering in detail each of the hormones and their mechanism of action. So let's start. So endocrinology is a subdiscipline of physiology. It focuses on the study of biochemical messengers and hormones. Hormones are basically secreted by endocrine glands, which are by definition ductless, meaning that the product of those glands are directly released into the bloodstream, and they get to their specific sites where they regulate very fundamental activities of the cell. And these are some of the activities that we will be focusing on in the introduction, but also as part of this whole endocrine physiology section. So as you can see in this slide, hormones really regulate and maintain the milieu interior, or the homeostasis of the cell. They regulate pretty much every organ system, and the hormones are the ones that hold everything together. In a very broad sense, hormones can be broken down by the biochemical composition. So they can be broken down as peptides, as amino acid derivatives, or as lipids. In terms of the peptides, which are by far the more common ones, a lot of the pituitary hormones, both the anterior and posterior pituitary hormones, come under this category. Other examples of peptide hormones are insulin, glucagon, or many of the growth factors like IGF-1, which is insulin-like growth factor 1. The second category of amino acid derivatives are, as the name suggests, are derived from amino acids like tyrosine or tryptophan. So tyrosine gives rise to a category of molecules called catecholamines, of which epinephrine and norepinephrine are members, and in fact also thyroid hormone, which is derived from tyrosine. In terms of tryptophan derivatives, the more common ones are serotonin and melatonin. And finally, the most complex of the three is the lipid class, and all of these hormones are derived from the backbone of cholesterol. So as you'll see in the adrenal cortex section, there are several hormones that are actually derived from the parent molecule cholesterol, which then give rise to many other hormones such as cortisol, aldosterone, or testosterone. In terms of how these hormones work, again, this is a general overview, and we will cover these again in more detail in the individual sections. But in a general standpoint, in terms of how peptide hormones work, using growth factor as an example, like insulin or insulin-like growth factor, what's shown in this slide is how these hormones actually bind to the receptor. And the binding to the receptor causes the receptors to come together, or what we call dimerization. And the dimerization then leads to a series of events, one of which is tyrosine phosphorylation. And the tyrosine phosphorylation in turn then leads and sets up a whole cascade of events called signal transduction that leads to further downstream events inside the cell that then signal the cell and also in the nucleus to lead to gene transcription, and then finally lead to a tissue response. And so just to put some context around this, for example, if insulin binds to its receptor, which is an insulin receptor, that insulin receptor has tyrosine kinase activity. So binding of the insulin to the receptor causes activation of the tyrosine kinase, and then that in turn leads to activation of downstream events, like for example transcription of GLUT4 or glucose transporter 4, which then sucks up the glucose inside the cell. We'll cover this again in more detail, but this is just an example of what happens. In terms of looking at the more specific actions of the peptide hormones itself, We just talked about tyrosine kinase, of which insulin is the prototypical example. But there are also other peptide hormones that work by working at another type of receptor called the G protein coupled receptor. In fact, the G protein coupled receptors form nearly 90% of the human genome. And so these are very, very common receptors. Many hormones bind to this class, and there are also many drugs that actually bind to this class of receptors. And listed in this slide is as basic list of many of the hormones that would be working by through activation of the G protein coupled receptors. So, for example, GS stands for the stimulatory subunit of the G protein coupled receptor, and as the name suggests, it stimulates or activates the second messenger cyclic AMP or CAMP. And some examples of those hormones that work by activating this receptor are many of the anterior pituitary hormones like ACTH or TSH or FSH or LH. And we'll cover again these in the anterior pituitary section in more detail. An example of a inhibitory type of G protein coupled receptor or GI receptor is the hormone somatostatin. And as the name suggests, this is working by inhibiting the second messenger CAMP. So there's a decrease in the CAMP when somatostatin binds to the GI subunit. Another category of G protein coupled receptors is a category called the GQ receptor, which actually works by a different second messenger pathway, which includes the second messenger IP3 or inositol triphosphate and DAG or diacylglycerol. So by activating IP3 or DAG, hormones that bind to this receptor lead to a specific action. And examples of those are the posterior pituitary hormones such as oxytocin and vasopressin, as indicated in the slide. So again, these are basic sort of concepts in terms of how peptide hormones in general work. They either work by activating the tyrosine kinase receptor, of which insulin is a really good example, or they work by binding to a G protein coupled receptors. And in the G protein receptor category, there's the stimulatory ones, the GS, the inhibitory ones, the GI, or a separate category called the GQ. In the next slide is a basic sort of schematic in terms of how steroid hormones work. So in contrast to the peptide hormones, the steroid hormones actually work by binding straight into, coming straight into the cell, because they are actually lipid derivatives, if you recall. And so they don't actually have to bind to a cell surface receptor. So these molecules actually diffuse easily into the cytoplasm. Then they bind to a protein inside the cytoplasm, as denoted by this PB. PB is basically the protein-bound version of the steroid hormone. And then they're in equilibrium, which then maintains the free hormone or the protein-bound hormone. And then the free hormone then binds to the receptor directly inside the cytoplasm or inside the nucleus, depending on what type of hormone it is. And then the binding of the receptor and the steroid molecule then cause translocation of that whole complex, the steroid and the receptor complex, by attaching to chaperone proteins called HSP, or heat shock proteins. So there are several heat shock proteins that basically chaperone or escort this receptor complex into the nucleus, and then binding of that complex to the DNA causes gene transcription, which leads to the production of an mRNA molecule, and an mRNA then that gets translated to a protein, and then the protein leads to a specific response. So again, this is left sort of very generic and general at this point to give you sort of an overview of how most steroid molecules will work. And in each of the sections later, we'll cover in specific detail how cortisol or progesterone or any of the steroid molecules specifically work. But this is the general scheme, where in contrast to the peptide hormones, which bind to the cell surface receptor, the steroid molecules are binding directly a receptor inside the cell. So that's the fundamental difference that you should keep in mind. Now we come to those, a little bit more detail in terms of how where these receptors are specifically located. So if you recall, the steroid molecule diffuses inside the cell to find its receptor. So the receptor can either reside in the cytoplasm, or it could reside in the nucleus. So those examples of the hormones that bind the receptor inside the cytoplasm are the ones that bind glucocorticoids, mineralocorticoids, or the androgens. So in other words, cortisol, aldosterone, and testosterone bind its receptor straight in the cytoplasm, whereas the hormones estrogen, thyroid hormone, vitamin D, and retinoic acid will bind the receptor inside the nucleus. So where the receptor is located inside the cell can actually dictate their mechanism of action as well. So let's test our knowledge in terms of just to try to put some of these concepts into practical use. So this question is asking, which of the following best describes an aspect of the mechanism of action of thyroid hormone? Is it A, binding of the receptors to the mitochondria? Is it B, binding of the receptors that directly modulate transcription of specific genes? Or is it C, which is the interaction and binding of a G protein with the plasma membrane? Or D, interaction with guanylyl cyclase without G protein? E, interaction with the plasma membrane receptor on target cells? Or F, participation by a second messenger? So the correct answer to this question is B, where the steroid molecule, in this case thyroid hormone, and if you recall again, thyroid hormone is a steroid derivative, they bind to receptors that directly modulate transcription of specific genes. Okay, so anything to do with CAMP or G proteins or any of those things, if you recall, these are activities of peptide hormones, not of steroid hormones. So that's a fundamental difference that you may wish to keep in mind. The next concept is the concept of feedback. And feedback is a very fundamental concept in endocrine physiology. And illustrated on this slide is the concept of a negative feedback loop. So negative feedback is somewhat like a thermostat. So if you set up the temperature to a certain degree, let's say you want to set it up to 65 degrees, and the outside temperature is 70 degrees, then the compressor kicks in and then cools the room to 65, and then once the temperature reaches 65, then the compressor shuts off. So it's kind of like that with the endocrine cell too. So the endocrine cells produce hormones, and the hormones work on target cells, and then once the specific level is reached, or a specific response is reached, then that response feeds back to the endocrine cell and shuts it off. And that shutting it off leads to finally a decrease in the production of the hormone. And that cycle is essentially a negative feedback loop. And basically illustrated here is a loop that goes from the hypothalamus to the anterior pituitary to the target gland, and then an effect. So the effect basically feeds back to the source and shuts off the response. And that basically, the decrease in the production is the concept of a negative feedback. In the next slide, we'll talk about the concept of positive feedback, which is exactly the opposite of negative feedback. So in this case, the target cell produces an effect, and then that effect actually feeds back to the endocrine cell, which produces that hormone, and results in further increase in its production, as opposed to a decrease in production, as we saw with the negative feedback. So a good example of this is the action of the hormone oxytocin, which is a posterior pituitary hormone. And the release of oxytocin from the posterior pituitary triggers uterine contractions. And this is a very specific event that happens during childbirth. So during childbirth, there is a need for increasing uterine contractions so that the fetus can come out. And this process is actually enhanced by a positive feedback. So the more uterine contractions that happen, that is the biological effect. that effect feeds back to the posterior pituitary to release more oxytocin, and then more oxytocin results in more contractions, and on that process goes until the fetus is expelled. So this is an example of a positive feedback. And it is important to note that most of these types of effects are happening during what we call climactic events, like childbirth or ovulation is another example of this. So that concludes the end of the introductory session. The main take-home points here are that we should focus on the feedback loops, especially the negative feedback, since we will come to this concept several times during the course of the lecture, and the fundamental differences between peptide and steroid hormones and their mechanism of action. In the next section, we'll dive right into posterior pituitary.